Hello, class. In this lecture today, we are going to talk about how we classify microbes. And so let's go ahead and get started with that. Um, After this section, you should be able to summarize the taxonomic hierarchy from domain to species, define the term strain, and describe the binomial nomenclature system and the information it provides about an organism. And so real quick, morphology and physiology are two of the primary ways that bacteria have been classified. So taxonomy is a study of how different organisms are grouped, and that's usually based on shared features. Now, originally that was based on morphological features like shape, size, and arrangement, um, and physiological features such as metabolism and like survival ability under different conditions. More recently, taxonomy has been going more towards genetic components, but we're going to just talk about some of the basics here. In this picture, all this cool stuff that you see are all the different types and shapes of microbes that have been found. Um, we often talk about a very minute portion of the shapes that are common or that exist in nature because they're specific to the kind of organisms that we can grow. But I'll tell you, my favorite is this little mustache in O. All right, so taxonomic hierarchy. There are 8 rankings within the taxonomic hierarchy. These rankings range from the broadest type, which is the domain, right? So we have 3 domains, the archaea, the eukaryotes, and the bacteria, to the more precise, which is at the species level. And so here we have them all laid out: domain, kingdom, phylum, class, order, family, genus, species. So the mnemonic here is delightful King Philip came over for great spaghetti. You may know more lewd and blue ways of remembering this, but this is the one that we'll use for class. Um, Now, what I will say here is that I'm not overly worried about you memorizing the order of these, um, but it is useful to kind of understand how they are grouped. So domain is the broadest category. It has the most members, while species is going to be a very specific organism. Now, our 3 domains are the bacteria, the archaea, and the eukarya. The bacteria and archaea are both prokaryotes. They are both unicellular. Um, and bacteria has some of our pathogens. Archaea, there are no known pathogens. They're pretty similar in where we'd find them, except for that archaea tend to live in extreme environments more often. Eukarya includes both unicellular and multicellular organisms, including ourselves. And so there are 6 kingdoms, which you may have heard about before. We've got the archaea, the bacteria, fungi, planta, animalia, and protista. All right. And so here they show up in the domain. The kingdom and the domain are the same for archaea and bacteria. And for eukarya, we have a couple of different kingdoms. The protist or kingdom protista, this is kind of a miscellaneous kingdom for organisms not categorized as, plants, animals, or fungi. But genetics now show that protists can't logically be lumped into a single kingdom. So really there's so much diversity here that there should be different kingdoms for them. And here we have an example of like a single organism following along their grouping system. So if we have this organism that we see here, which is Clostridium tetani, they're in domain bacteria, which is kingdom bacteria. They're in the phylum Firmicutes, class Clostridia, order Clostridiales, and the family Crostidiaceae, genus Clostridium, species Tetani. And when we talk about an actual organism, we in their name, we go with this binomial nomenclature, which we'll talk about here in a moment. Now, um, there are two different ways of grouping species. In the eukaryotic sense, these are a group of organisms that can sexually reproduce together to produce viable offspring, is the important part that I don't have on there. Prokaryotic species is a little bit different. They don't sexually reproduce. These are cells that share similar physical characteristics and have at least 70% DNA similarity. And then we also use um, what is known as 16S rRNA sequence similarity. And for species to be the same, or to be within the same species, we usually consider that 97% similarity. And within the same strain would be about 99% similarity. So strain is something that's kind of specific to prokaryotes. Um, it's used to recognize genetic variants within the same species. And so you can get mutations in a species that leads to a different strain. Now, oftentimes these are delineated by numbers or letters. For example, we have Escherichia coli, which comes in forms like E. coli K12 or E. coli O157H7 or E. coli B or E. coli C. And so E. coli K12 is the one that we commonly use in the lab. Now, the primary way that we name organisms at the species level is we use binomial nomenclature. Binomial basically means two names. This is the genus, and then we have the species. The genus is going to be uppercase or capitalized, and then the species is lowercase. Scientific names are underlined when handwritten or italicized when typed. Just keep that in mind. And just as something, because this will come up occasionally, the plural of genus is genera, and the plural of species is species. An example of binomial nomenclature is Escherichia coli or E. coli. A lot of classification comes down to using Berge's Manual of Determinative Bacteriology. It's got a list of all these different organisms, and you can follow through based on its physical and biochemical characteristics to determine its actual species. Morphology is one of the most common ways to start your classification. The big ones are cocci and bacilli, which we will, see in class. Then you have some others that are kind of common. You got these variations on the rod, like the vibrio or the curved rod, or you have these helical rods. You have these spirochetes, which are little spirals. You got these really long rods that are filamentous. You get these club-shaped rods, and then you get these other different types of spirals. You get some organisms that have these appendages, like these hyphae and stalks. And so these are the ones that we see most commonly in the lab setting, but as I mentioned earlier, they come in a great variety of shapes. So these are multiple, these are a bunch of the different shapes that have been found in bacteria that you can see that the diversity is much greater than what you get in just cocci, bacilli, and spirals. And so that is the end of this section. We'll talk about some of this material more as we go on, but at this point, you should be able to summarize the taxonomic hierarchy from domain to species, define the term strain, and describe the binomial nomenclature system and the information it provides about an organism. There is an objective check loaded in D2L, so please take that short mini quiz and determine if you have met these objectives. All right. Thank you all, and I'll see you in class soon.